So immunotherapy is a type of therapy that literally is on the forefront of science. There's new developments in immunotherapy all the time. Some great successes, other times maybe not as great successes they expected, but it is very, very current. So basically with immunotherapy is we're helping your immune system fight disease. A lot of times we're talking about cancer if we're talking about immunotherapy, but there are other illnesses that can be used, that can be treated using immunotherapy. So immunotherapy has a bunch of different names. I've got a couple of them here. So you may hear people talk about biologic therapy, using biotherapy. You may hear them being treated with biological response modifiers, or you could hear them just being treated with people being treated with biologics. So anything like that are all immunotherapy. And there's other terms that are used to describe immunotherapy as well. So there's a couple major types of therapies. The first off is passive. So remember, what we're going to be doing here is we want our own immune system to be doing the job. So what we're going to do with passive is basically supplement your immune system with human-produced cell proteins. So we're going to make cell proteins in the lab, and we're going to give them to the person to help supplement the immune system that they already have. There's no memory response with this, so it's not like you're going to be building up an arsenal of cells to go and attack something, but it actually happens quite quickly. So that's one good thing about them as well. Now with active therapy, what we're going to be doing here is we want to stimulate your immune response that's already in your body. So with the first one, we're going to supplement your immune system with new proteins. And with this one here, what we're going to do is kind of slap your immune system and get it to wake up and do its job properly. It forces our own antibodies to get working, so it's going to, get your own self to produce antibodies so that you do have a memory response to it. And this one also happens a lot slower because your body takes time to make antibodies. They don't just, make antibodies all of a sudden. Now we can also take these two types of therapies and kind of mix them together to form a hybrid, which is a combination of active and passive therapy. Alrighty, what I'm going to do here is we're going to go through all the different types of therapy, and then tomorrow's note will be the diagrams that go with this. If we were doing this in class, I would have, notes, and then we would do it in colored chalk on the board, and it would take us a couple days, but I've just changed it a little bit so we can get through the written stuff today and then the drawings for tomorrow. All right, so the first one we're going to talk about are monoclonal antibodies. This is one you hear a lot. Now, monoclonal antibodies, remember the short form for antibodies is AB, so big A, little b, and then monoclonal, I've underlined these for a reason. You'll see a little bit later on, okay? So just to help you remember monoclonal antibodies. So this is a form of passive immunity. It is one that's most widely used, which is why I just said this is one you've heard of a lot. So what we're going to be doing in the lab is making antibodies in the lab. I remember when my husband was in undergrad, one of the classes he took, I didn't take it, was, I can't remember, some immunotherapy one, and they were actually trying to make monoclonal antibodies. So this was a long, long time ago, and the lab they kept getting contaminated. So it's really neat that they were struggling to make the monoclonal antibodies, and now they're being used widely in drug treatments this many years later. So basically what these guys are going to do is they're going to boost the immune system function. And whenever you see a drug that ends in MAB for monoclonal antibody, you know that that is going to be a monoclonal antibody treatment. So basically we can use monoclonal antibodies to treat a number of diseases. So we can use them for leukemias, we can use them for lymphomas. This is where I'm saying that sometimes you can use them for diseases that aren't cancer. So they use them for lupus, for multiple sclerosis, and a lot of times you can use them for rheumatoid arthritis as well. So they've had great success with treating some of these illnesses, well, all these illnesses, but some of these non-cancer illnesses with these monoclonal antibodies. Now, there are side effects with using monoclonal antibodies. The people can have a huge allergic reaction to them. And with any type of drug, remember as well, this goes for all of them, that you can have side effects. Now, another one here are immune checkpoint inhibitors, or ICIs. So these ones are really neat as well. I think you're going to really get a nice visual of them when we do the diagrams. Kind of makes I don't know, I think this stuff's really exciting. But anyway, basically what, this is another passive form, and what checkpoints are. So on your immune cells, there are two switches. Well, there's more than two switches, but there's two basic switches. There's an on switch and an off switch. When the on switch, when the protein that is on is stimulated, it will turn the T cell on. So the T cell will go around and it will start to kill foreign things. So that's if that switch is stimulated or activated. If the off switch is activated, it turns the T cell off. And cancers are very sneaky because what they can do is they can stimulate or activate the off switch. So they literally turn the T cells off, so they're not doing their job. So what we can do is we can block those off cells from the cancers being able to get in. So the cancer cells manage to turn off the immune cells. So what the checkpoint inhibitors do, I don't know why they call them checkpoints and not switches, but whatever, is that it blocks the off signal. So what I say, it's like putting glue into a lock so you can't put the key in there. So the cancer cell has the key that fits in the off switch, because cancer cells are really sneaky, and of course they've stolen the key from somewhere. So what we're doing is we're putting glue in the lock so that they can't put the key in the off switch. And they've got some really good results using ICIs for melanoma, for a lot of head and neck cancers, and also cancer vaccines are similar to how we just use regular vaccines. So what we're going to do is we're going to present the body with what the bad guy looks like so that our bodies can produce the immune cells that they need to fight those bad guys. So this is an active form of immunotherapy. It helps the immune system launch an immune response to a certain disease. So we're going to show the body what this disease looks like so that it can produce antibodies against the disease. What we're going to do is we're going to take the cells from the tumor, and we're going to take a protein from that tumor, and we're going to add a little protein on it to make it look more recognizable. So if you remember back to your vaccines, when we had things like the pneumonia vaccine, which had the polysaccharide around the outside, we said children's bodies don't recognize the polysaccharide very well. So what we need to do is make that polysaccharide more recognizable. So we put a harmless protein on the end of that polysaccharide, and the children's body was like, oh, I see that polysaccharide is linked with that protein. That's actually a bad polysaccharide. We want to kill that thing. So we're going to do the same thing here. We're going to take a piece of protein from the tumor cell, add another little piece of protein on it to make it more recognizable, and then your body is going to go, wait a minute, I recognize that protein because it's got that little marker on the end of it. That's a bad guy there. So basically that's what we're going to be doing here. Same thing as with our vaccine that we made. So we're going to take that protein that we made more recognizable. We're going to inject it back into the patient. So now it's got a little flag on the end. So when the patient's body see that protein with this little flag saying this is a bad guy, it's going to know to make the proper T cells to recognize the cancer cells, and then those T cells can go in and kill those cancer cells. Most of the vaccines for this cancer vaccines are in clinical trials, but they have had really good luck doing some cancer vaccines for prostate cancer. Alrighty, next one. So for non-specific immunotherapies, this one's a little bit broader. Now, this is also a form of active immunotherapy. And in this case here, we're not going to target the actual immune cells. So if you're just walking around, you're all healthy, everything's fine, you got some lymphocytes going around, you got some neutrophils going around, just make sure everything's okay. As soon as you are sick, all right, as soon as you've got some condition, what ends up happening is those cells that are just kind of, circling around making sure everything's okay, they go into attack, but at the same time, they also release chemicals or chemical signals that tell the other immune cells, come here, I need help, hurry up, multiply, we need more of you, come on, hurry up, hurry up. So what non-specific immunotherapies are is they're basically the chemicals. You're going to give those chemicals to the patient, and those chemicals are going to signal the patient's immune response or the immune system to hurry up and make some more of those cells that can come in and do some killing. So they don't target the actual immune cells. They don't, you're not putting T cells or anything into the body. What you're doing is you're putting the chemicals that tell the other cells to hurry up and multiply and get over here and do your job properly. So we've got those immune chemicals that are made in the lab, and they will be injected into the patient, and those chemicals will signal the immune cells. So they basically stimulate the immune system to work harder. So there's just more of those chemical signals saying, come on, hurry up, hurry up. It's like instead of one person yelling at you to go fast, you have a whole crowd of people yelling at you to go run faster or whatever it is, or kill more. So for example, the cytokines. So things like your interferons are important to decrease the growth of cancer cells. And then the interleukins are important to increase the ability of white blood cells to kill cancer cells. And this is used for things like, again, your melanomas, and for some of your kidney cancers. And let's see if there's one more here. Oh, yeah, and there's your bladder cancer. Earlier on, I thought bladder cancer was one of the earlier on slides, which is why it ended so abruptly. So these are the kind of cancers that we use immunotherapies for, or non-specific. So our last one here is CAR T cell therapy, and the CAR stands for Chimeric Antigen Receptor. What we're going to be basically doing here is we're going to be taking some T cells, and we're going to be putting some proteins on the outside of their T cells that come from elsewhere. So that's why it's got the name Chimeric. This one's, again, I was looking at this last night, and it hasn't actually been approved in Canada as of now. But it is approved for some cancer therapy in the United States. So you'll hear about a lot of Canadians that go to the United States to get CAR T cell therapy. Mr. Hopper's little niece there, that's what she was doing, was going to Philadelphia to get CAR T cell therapy for her leukemia. So this is a hybrid. I got the star next to it because I was looking stuff up just to know exactly which category to put it in. And one place would say it was active, one place it would basically say nothing. And then another one would say, well, it's an active with a form of passive. So I put it under as hybrid because I think that's that is what it basically is. So it's got components of both. So basically what we're going to do is take the T cells out. So we've got T cells in our body. Remember, they're the ones that go around, they learn what the bad guys look like, they go in there and they attack them. So we're going to take those T cells out, because sometimes with some cancers, like we said earlier, they're turned off or they can't recognize what the cancer cells look like. So we're going to take those T cells off, and we're going to teach them what the cancer cells look like. So we're going to engineer them in the lab to recognize cancer cells. Then we're going to inject them back into the patient, and then those modified T cells, so the ones that we've just taught to know what the cancer cells look like, can go in there and attack the cancer cells. So like I said, they're currently in clinical trials, but they're used for acute lymphatic leukemia, and it's okay for that in the United States for this disease. So tomorrow when you tune in, we will be doing diagrams for each of these types of immunotherapies. So get your colored pencils out, and or pencil crayons as they say up here in Northern Ontario, and we'll do this. Okay, talk later.